Can somebody confirm you can hear me?
I can hear you.
Thank you. All right, before I share my screen, let me talk about the grading. Let me ask before I begin, are there any questions on anything? I think I need that, so let me close that down. Sorry, my computer is working really slow. It's because the lecture file is still being made on my computer. All right, I got the extra study sheet graded. I think I got that graded before. I got the extra credit picture graded. I got the extra credit study 2 graded. Worksheet lecture 3 is graded. Student discussions are graded, except maybe one student. All of the quizzes have been graded. The last quiz being quiz 1C. And somebody's already taken quiz 2C. How is that possible? I thought it didn't open until midnight tonight. Let me take a look at that.
It posted for midnight, like this morning AM, all the
All
way
right.
until
Yeah,
tomorrow
that's not what it's
midnight.
supposed to be. It's supposed to be midnight tonight. All right, I'll have to change that.
Just so you're
Obviously,
aware, all of the quizzes I think so far have been posted on Tuesday morning and then due by Wednesday night.
yeah, it's supposed to be Tuesday night, meaning 12:01 a.m. And you're saying they're posting, well, 24 hours earlier. You're not supposed to have 48 hours to do this. You're supposed to only have 24 hours. So I must be not noticing that it's taking a different time than I thought. I thought it was going to be 12:01 a.m. Apparently, it's 12 midnight the day before. All right, I'll take a look at that. Thank you for letting me know. Um, I never noticed that earlier, but there it is there. Anyways, quiz 1C. I don't know if there's any extra credit questions. I got the environmental scan graded, and about 90% of you did that very well. There was one student I gave a warning, and then one student I um, I think I penalized them because they didn't give me the environmental scan. And then the worksheets for Lab 0, Lab 01, Lab 2, Lab 3, and almost Lab 4 are graded for one time. Some of them for two times. I don't remember which one, but probably Lab 2 has been graded two times. But Lab 3 and 4 are almost graded. Lab 3, everyone was graded one time. Lab 4, I've got four more students to finish. I'll get that done tonight. And then Lab 4 will be graded as long as I get it done by Wednesday morning. And we'll say just early Wednesday. Uh, the Lab 3 and 4 will be due for the second grading this Saturday. Remember, in the first grading, I'm only going to tell you what's wrong. I'm not going to give you what the answers are. If you want to know what the answers are, you either have to turn in the lab again, and either you'll get it right the second time or wrong. And if you get it wrong the second time, I'll tell you what the correct answer is. If you only turn in the lab one time and you want to know what the answers are, you need to email me, and then I'll send you what the answers are. Any question about any of that? All right, I think that's it. So that's almost everything in the class is graded, except for the four students of lab 4. And that isn't even due yet, so I'm not going to grade that. The only thing that might not be graded is extra credit study 3, and I'm not sure that's due yet. Let me look that one up. Yeah, that one is due, so I can get that one graded. All right, so everything is graded except for extra study 3 and the 4 people in lab 4. Any questions about the grading?
I just wanted to ask, is there a reason why the quiz that we only have 24 hours to do it and it's due on a Wednesday?
Well, Wednesday was the what the students voted for a long time ago. People didn't like having the quiz on the same day as the lesson because some people would be working and then come home to the lecture and they'd just be tired to take the exam in the evening. And I said, that's fine, we can have the quiz on Wednesday. Um, it has to probably be on a Tuesday or a Wednesday. You guys could obviously vote on that if you don't like it on Wednesday. The quiz open for 24 hours was a decision made by the biology department at Clark College. It is not my decision. Okay. And they decided to have the quiz open for only 24 hours to reduce on student cheating. Okay.
Okay, that makes sense.
All right. All right. I shut down the grade book. Let me open up the share my screen. Let me see if we're still running Zoom. I think the Zoom from the lesson, yeah, for the lecture, it's finished. All right, good. So let's go to the lesson. Today is January 27th, and we are going to be covering Lab 5, Streaking for Isolation and Culture Media. You do need to read the unknown project instruction files on Canvas for next week. I'll remind you on Thursday, but you can find the unknown project instructions in week 5 of the lab. So there's week 5. Whoops, I lost it now. I had it. Week 5 lecture, week 5 lab. Unknown reports, the main part of week 5 lab. The unknown instructions. You need to have this read by next week. Any question about that? The other stuff I'll talk to you next week. You don't need to have that read yet, but we'll talk about the unknown project next week. Any questions about what we're going to do? All right, let's move to Lab 5, cultivation of bacteria, culture media, inoculation, and isolation. Like all of the labs, there is some pre-reading you should do before you do the lab. Not too many pages. What is that? Less than 5 pages, maybe 4 pages of culture media, and then a page on obtaining pure cultures. There are some video clips in today's lab. The virtual lab no longer works, so it's been removed. It was using Flash, and Flash no longer supported and no longer works, so it's been removed. The learning objectives for this lab: 1, understand the importance of proper specimen collection and transport to the lab. 2, explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between various types of culture media. 4, explain the purpose of streak plate culture technique, aka streaking for colony isolation. And then 5, define the following terms: selective media, differential media, and reducing media. Any questions about these objectives? All right. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they can dry out, or exposure to oxygen. Almost all anaerobes do not want to be exposed to air or oxygen, and they therefore require a special transport medium. Actually, almost everything requires a special transport medium. Once a specimen is received in the lab for a microbiological culture, microbiologists employ the 5 principal I steps to process it. 1, inoculation; 2, isolation; 3, incubation; 4, inspection; and 5, identification. So 1, inoculation, that's placing the specimen on an artificial culture media to initiate growth of the culture. And then you go to 2, isolation. Once you have it grown, you start spatially separating out microbial growth into distinct colonies. You incubate the culture by placing the culture medium in environmental conditions to facilitate growth. Usually, you incubate a culture in an incubator. Let me show you what an incubator looks like. We have incubators in the microbiology lab, if you ever were to go into it and take a look at it. Oh, come on. Maybe I went past it and didn't find it. Up here we go. There's an incubator. So you isolate out the growth, get isolated colonies, grow up pure cultures, and then you inspect the culture, examining the culture for the presence of microbial growth. You can make some determinations from what you view, but you're not going to be able to identify it just from these steps. You then have to go through identification. This is usually done by performing specific tests to ID any pathogenic microbes that are isolated. We'll talk about some of the different tests you can do, beginning with um, this lab here. And of course, if you had a really fancy lab, you could even sequence the DNA and find out specifically what that one pure culture is, colony is, and growing up to get a pure culture. Most microbiology labs do not sequence the DNA, they just run biochemical tests to identify the specimen. The 5 steps will be described in detail after we discuss different types of culture media. You're already familiar with general purpose media. We've already talked about Na, nutrient agar, Na, or nutrient agar with yeast extract, and then TSA, or tryptic soy agar. These are general purpose media to grow many organisms cheaply. There's no selection. Not all organisms will grow, but many will. And general purpose medias are just cheap and easy to use. The organisms that will not grow on a general purpose media, we call them finicky organisms or fastidious. And you need to add specific nutrients to get the fastidious microbes to grow. To get a fastidious microbe to grow, we generally grow them on an enriched general purpose media. One such general purpose enriched media is blood agar, which has 5% sheep's blood simply added to TSA. And then you can grow many fastidious organisms, like Streptococcus pyogenes, will not grow on a general purpose media, but it will grow on blood agar because the blood cells supply the nutrients that Streptococcus pyogenes requires in order for it to grow. Any questions about any of that? So blood agar, abbreviated BAP for blood agar plates, or chocolate agar, CAP or CAP for chocolate agar plates, are two enriched general purpose media. And these are probably the two that you'll be most likely to run into in a microbiology lab. But you could have other enriched general purpose media where you have like TSA, and then you add specific additives to it to get certain species of bacteria to grow. Any question about any of that? We also have selective media, and these are media that contain chemicals to inhibit or halt the growth of some organisms. So some organisms can grow on a selective media, and other organisms cannot. Keep in mind two things when you're using selective media. First, the selective media isn't necessarily perfect selection, meaning if given enough time, even organisms that are inhibited may form visible colonies. And second, even organisms that are supposed to grow on the selective media may more form smaller colonies or grow a little slower on the selective media than they would grow on a general purpose media. One example of a selective media is 7% sodium chloride. It has 7% sodium chloride in the media. If the organism likes growing on 7% sodium chloride, it will be selected for. If the cells do not like or do not grow on 7% sodium chloride, they will not grow and will be selected against. Most bacteria will not grow above 2% sodium chloride. So they will not grow on 7% sodium chloride media. And an example of that would be, well, Streptococcus pyogenes, grown on blood agar. If you were to add 7% sodium chloride, it would not grow. An organism that would grow on 7% sodium chloride is Staphylococcus aureus and Staphylococcus epidermidis, as well as Micrococcus luteus. These are organisms that can grow on human skin. And as you don't know, the human skin can become up to 20% sodium chloride. And obviously, these organisms can grow up to 7% sodium chloride. Not 7, 20% sodium chloride. So they can easily grow on 7% sodium chloride. Any question about any of that? All right. We also have differential media. These media contain chemicals that produce visible differences among different groups of organisms when they're grown on that medium. These differences are unrelated to how well the organism can grow on the media, but they usually allow you to visually look at the colonies and see differences. Like the colonies could be different colors, they could have a different aurora of the agar around the colony. Anyways, the differences allow you to see differences among dissimilar bacteria by visually distinguishing the two colonies from each other. An example of a differential media is blood agar, which allows you to distinguish between cells which can hemolyze the red blood cells as to cells which cannot hemolyze the red blood cells. And you can distinguish between three different types of organisms. Let me blow this up a little so you can see it a little better. In the gamma here, we can see the red blood cells stay red all the way up until you have the bacteria cells, and then really you can't see the red. And this is gamma hemolysis or no hemolysis of the red blood cells. We also have alpha hemolysis, and that's shown here in the alpha, where the bacteria are the off-white color of the alpha. And you can see the red, and right before you come to the off-white of the alpha, we have a darkening of the blood. Inside the alpha, the entire area is dark. That darkening is from partial hemolysis of the red blood cells, making the agar either look dark or greenish. Sometimes it looks like a greenish tint. And that's from partial hemolysis of the red blood cells. You can also have beta hemolysis or complete hemolysis, where all of the red blood cells have been lysed. So the white beta shown here is our streak of bacteria. And then we have a clear region around it where all the red blood cells have been lysed. Inside the beta, all the cells have been lysed. Around the beta, all the cells have been lysed. And so we have a clear region. You can actually hold this plate up to the light and you can see through the beta hemolytic areas. Like I can shine it and see the light bulb of the light, or you can shine it up to the clock and tell the time through the beta hemolysis. Around the beta hemolysis, you usually have a dark ring, which is alpha hemolysis, and that's normal. You usually find alpha hemolysis outside of beta hemolysis. Not terribly important. And then out further, you have normal red blood cells, meaning no hemolysis. Anyways, the blood agar plate allows you to visually differentiate between the different types of hemolytic activity. So blood agar is a differential media plate. Any question about any of that? All right, you might notice that blood agar was an enriched media plate. It is also a differential media plate, and you can have plates with more than one category. One other plate, which is both differential and selective, is McConkey's agar. If the bacteria can grow on the McConkey's agar, then it is gram-negative because the McConkey's agar selects for gram-negative organisms. So this organism is gram-negative, this organism is gram-negative. It can grow on the McConkey's plate. I don't have a picture of an organism not growing here. If I scroll down, maybe I got one. Nope, I don't. Anyways, there's no growth. It's gram-positive, and that's because McConkey's is actually pretty good at selecting for gram-negative, meaning 95% of the cells that grow, meaning cultures that grow on a McConkey's, will be gram-negative. And if it does not grow on McConkey's, then it's gram-positive. And like I said, it's good with about 95% or greater of the species of gram-negative and gram-positive. And in addition, it's also very good that the selection tends to be 100%, meaning the cells will either grow or they won't grow. There's very few specimens that'll grow slowly on McConkey's. It is also differential in that you can differentiate the cells on their ability to ferment the sugar lactose. If the bacteria can ferment the sugar lactose, you will see that the colonies will appear pink, and then the agar around the colonies will be pinkish as well. And that's because the lactose fermentation puts out acidic end products, which will change the color to pink of McConkey's. If the bacteria cannot ferment the sugar lactose, then the color will be the normal color of the bacteria on the agar. In this case, the bacteria is an off-white color, and it's not pink because it cannot ferment the sugar lactose. So non-fermenters will be some other color than pink. Fermenters will always be pink. Any question about any of that? So McConkey's is both selective and differential. I shrink that down. Lastly, we have a reducing media. These media promote the growth of anaerobic bacteria because they have low or no oxygen conditions in the tube, which is required to grow anaerobes. It's important to note, I guess I already stated that the media can be classified into more than one category. All right, this table is showing you the different types of media. We have a general purpose media, and we've already talked about them in the previous lab. There's tryptic soy agar or TSA, nutrient agar or NA, or nutrient agar with yeast extract or NA, and it's all a general purpose media promoting the growth of most organisms, but it does not promote the growth of fastidious organisms. To grow a fastidious organism, you have to have a enriched general purpose media like blood agar or chocolate agar, BAP or CAP, which can grow a fastidious organism like Streptococcus pyogenes will grow on blood agar. Haemophilus influenzae, which is a fastidious organism, will grow on chocolate agar. And what happens is both of these plates have blood agar added to them. The blood agar has the blood added after the agar is cooled to, I think it's 50 degrees or less. And then you add the blood and then mix it around and then pour the plates while it's still liquid. And the chocolate agar, you're adding the blood to the agar when the agar first comes out of the autoclave. So it's very hot, close to 121 degrees Celsius, and that will lyse the blood, causing the blood or the media to take on a brown appearance, which the brown looks like chocolate, but there's no chocolate added. It's actually blood that's added. Remember, blood agar is both an enriched general purpose media and a differential media. So it's enriched because you can grow a fastidious organism like Streptococcus pyogenes, and it's differential because you can differentiate between the hemolytic activity of an organism growing on blood agar. Chocolate agar is only enriched general purpose media because it can grow a fastidious organism, but it is not differential. And then we have McConkey's, eosine, methylene blue, and hectone enteric agar, also called MAC, EMB, or HE agar. These three are all selective and differential. They are all selective for gram negatives, and they select against gram positives. And they're all differential on the basis of lactose fermentation. If the organism can ferment the lactose, there'll be a different color. On McConkey's agar, it will be a pink. On EMB, it'll either be a dark color like black or a metallic green color if it can ferment the sugar lactose. And on hectone enteric agar, if it can ferment the sugar lactose, it will be a yellow-orange color, which is called salmon colored. We also have phenyl ethyl alcohol or PEA. It is a selective media. It has alcohol in it. Phenyl ethyl alcohol is the alcohol, and it is selective against gram negative. So it selects for gram positive organisms. The PEA, the phenylethyl alcohol, gets in the gram-negative cell wall and prevents the cell from growing. And so the gram-positives can grow on PEA, the gram-negatives cannot. We also have mannitol salt agar. It is selective in differential media. It is selective because it has 7% sodium chloride in it. The organism will not grow unless it can tolerate and grow in 7% sodium chloride, like Streptococcus pyogenes would not grow. What's another organism that would not grow? Oh, E. coli would not grow on mannitol salt agar. Staphylococcus species like Staphylococcus aureus, Micrococcus luteus will grow on mannitol salt agar because it can tolerate the salt, so they're selected for. Mannitol salt agar is also selective, differential. It is differential on the basis of the ability of the species to ferment the sugar mannitol. If the bacteria can ferment the sugar mannitol, it will change the pH, meaning acids will be released, and the agar around the colony will take on a yellowish appearance from the increase in the pH. So that's how mannitol salt agar is differential. If the bacteria cannot ferment the sugar mannitol, then the agar around the colony, meaning the aura of the colony, will not be yellow, and the plate will be its normal color, which is a kind of a pinkish color for mannitol salt agar. All right, there's isolation, growing the culture that you got from a patient in a dish. You then would have to separate out the colonies. Like you could take that colony here, take a swab of this colony, streak it out on another plate to get isolated colonies. Take a isolated colony from that second plate, and then grow that up and call that your pure culture. Remember, you have to get cells through two isolated colonies before you can call it a pure culture. And even then, it's only about 95% or maybe a little higher chance that it's a pure culture. You could still not have a pure culture, meaning you would have to streak it out again to get isolated colonies to get a pure culture. Let me move that picture down a little.
I'm so sorry. Can you explain one more time how you're able to mix them to create a pure culture?
We just got a specimen from a patient which has many different organisms in it. Some of them will be normal microbiota. Like if this is from the skin, I don't know where this is from, but that looks like it could be just a mold from the skin or yeast from the skin. This looks like that one there could be Staphylococcus aureus, for example. Those, I don't know what would be white from the skin. A small yellow colony, oops, sorry about that. A small yellow colony, like maybe that one, could be Micrococcus luteus or that one there. And so you just obtain cells from the patient and they're going to be mixed. You then have to start isolating out the cells and the species from one colony. It only is going to be about 70% pure, meaning this colony here could be 70% pure, but then 30% of the time it would not be pure. And you streak it out, you'll get mixed colonies growing on the second plate. And so you have to take from an isolated colony from the second plate to get out the pure culture from this colony. Any question about any of that? Or did I answer your questions adequately enough?
Okay, so pretty much just to clarify, you're taking one plate, streaking it from one of those colonies, and then just repeating until you have it isolated.
Correct.
Okay.
Now, obviously, on this one here, we have some isolated colonies, like this one here and these colonies here. But see that one there, and this one there, and that one there? These are not isolated colonies. So if you needed to grab from this one, that's not from an isolated colony. So you have to take it from here, streak it out on the first plate, and then that's get one isolated colony. That's the first time you sent through an isolated colony. And then you have to take from that plate and streak it out on a second plate to get a second isolated colony. But my point is, you have to have isolated colonies two times, at least two times, before you can call it a pure culture. And then, even then, you're not guaranteed that it's pure. There's never a 100% guarantee that it's pure, but you can state that it's at least 95% pure or greater if you sent it through two isolated colonies. Now, that one there is not an isolated colony because two colonies are together. Any question about any of that? All right, let's move on. Isolation. Louis Pasteur developed the concept of obtaining a pure culture. His way of doing it was very difficult. He had to grow up the culture in a liquid broth, and he made dilutions of that culture, different dilutions like 1 to 10, 1 to 100, 1 to 1,000, 1 to a million, whatever. And then he replica plated each dilution. If he ever got one dilution where one tube had growth in it and nine tubes had no growth in it, he correctly surmised that the one tube with growth in it, out of the nine tubes that did not have growth in it, that dilution was a pure culture. It was a very difficult way of getting a pure culture. Uh, Dr. Koch came up with an easier way, and that is you just streak the cells on a solid media, streaking out for getting isolated colonies. Take from a single isolated colony, streak it out on a second plate to get isolated colonies. And then you take cells now that have gone through two colony isolation events to set up your pure culture. And even then, like I stated, you're not guaranteed 100% that you have an isolated colony, but you're guaranteed that you have at least 95% or greater that it's an isolated pure culture. Any question about any of that? You don't need to know the time when they did that, but you should know that Louis Pasteur first developed the idea of working with the pure culture, and he had a very difficult way of obtaining a pure culture. Dr. Koch came up with the streak plate colony method, or streaking for colony isolation. And if you do streak it out two times for getting two isolated colonies, then you can state with 95% confidence or greater that you have a pure culture. And how you streak it out on a plate, this is actually what somebody did streaking out, is you take from your stock culture, streak it out in quadrant one, sort of like this. And in reality, you don't streak it out. What is that? One, two, three, four, five or six times across. You want to streak it out 20 to 30 times across sector one. Okay. And it's fine if you overlap, like this person there overlapped right there. And then you sterilize your loop. So I'm going to pretend this is a loop by putting it in the back incinerator or passing it through the flame, cooling the loop down. Usually, what you do to cool it is you touch it on sector two, someplace where you're not streaking, like right there. And then on sector two, start streaking on sector two, going into sector one in the low portion of sector one to streak out sector two. So in reality, this is not a good one because this one is going here and there. On sector one, this is the high density of sector one, this region here. you never want to go into the high density. You only want to go into the low density. And as you're streaking out sector one, you'll get fewer and fewer cells the further you streak. So the lowest density will be here, the highest density here. You never, when streaking out sector two, go into the high density. You only go into the low density. So the person should be streaking in sector two, going to about here, and then stopping, going back to sector two. Let's go down to here. And then coming back into the low density of sector one and going back into the low density of sector one 3 times. They did that a little lot more. They did 1, 2, 3, 4, 5 times. You only need to do 3 times. And then streaking it out on sector two. Once again, you streak it out closer to 20 to 30 times, not, I don't know, 6 to 8 times. And then you sterilize your loop in the flame or Bechtel incinerator. Touch it to the, let me use the mouse again so you can see it. Touch it to this portion of sector three. And then this one, it's showing it correct. You come into sector three, streak into sector two. And this one's almost right. Only go in the low portion of sector three. Streak out, going in once again 3 times into sector two. You do not want to go into the high region of sector two, this region here. You only want to go into the low region of sector two, this region here. And then streak out sector three 20 or 30 times. Then sterilize your loop. And this one, they actually did it right. And streak, cool the loop out by touching it on sector four. Bring your streak into sector three, only going in the low region, which is only this region here. Go in about 2 or 3 times. And actually, this one, they did it right. They only went into the low region, they didn't go into the high region. And then streak out on sector four, and you want to streak it out about 20 to 30 times. You can call this quadrants if you're using 4 sectors. You can also call it sectors, and sometimes you only streak it out 3 times, in which case it would be called the sector. You then get an isolated colony. Take from an isolated colony, streak it out on another auger plate, streaking out for colony isolation. You could use the 4-quadrant method to get the isolated colonies. And then to get your pure culture, you take from the second plate, take from cells that have gone through 2 colony isolation events. On the first plate here, and then on the second plate here. And then you set your stock culture up from the cells from an isolated colony on the second plate, meaning from cells that have gone through 2 colony isolation events. Any question about any of that?
Just to be clear, when they're streaking, they are not grabbing new bacteria from like their original sample. They're just taking it from each of the sectors and dragging what did grow or what you did streak and
Yes,
dragging it along.
yes.
Okay.
You have to go from the sectors because you want the bacteria in sector one to be the heaviest, and then in the last sector to be the lightest. And someplace along the line, you will have isolated colonies forming. Here we're getting it in sector four, but if we have fewer bacteria, or if they'd done a better job of not going into the high density of sector one and sector two, you could get isolated colonies in sector three, or even maybe in sector two. It doesn't matter where you're getting isolated colonies, but you need to get isolated colonies to get a pure culture. Although you need to streak not from one isolated colony, you need to then pick from this one isolated colony and streak it out a second time. If you were to set it up from your stock culture for each sector, you would have no dilution. Each sector would have the same concentration of bacteria. And then you would never get isolated colonies. They'd all be like sector one. Any question about any of that? All right, for those of you who don't know, there's actually a question about that in this lab. All right, so then we have incubation. The culture media contains chemicals and additives, like the nutrients necessary for the microbes to grow, but we also have to put it in physical environment for the microbes to grow. You can have a sophisticated incubator, which we do not have in the microbiology lab at Clark College, which actually can control how much O2 and CO2 will be exposed to the microbes. For example, if you're going to grow, all right, I just thought of one, Helicobacter pylori, you'd want to grow it at higher CO2 and less oxygen than you find in the normal air. Because Helicobacter pylori grows in your stomach, so the oxygen level will be something like 2 to 4%, and the CO2, I don't know what it would be, but it would be higher than what is 0.04%? Is that CO2 in the air? Whatever is in the air, Helicobacter pylori will like it higher than CO2 than the air because the stomach has higher CO2 than you find CO2 content in the air. Another example would be gonorrhea. If you're growing that bacteria up, it prefers a little less oxygen, like maybe 10% oxygen, and a little more CO2 than you find in the air. And so you'd want to grow that in a special incubator where you could control the amount of oxygen and the CO2. Our incubators only control the temperature, the humidity, and the time. The time is you put it in the incubator and then you allow it to grow until you pull it out of the incubator. So you determine how much time. The humidity in the incubator is present by, you see that container there? You put water in there, and then the heat of the incubator makes the water evaporate, bringing humidity into the air of the incubator. That's important because if you have a plate, like right there, growing in the incubator, the plate can only survive for about a week if you do not have humidified air. With no humidity in the air, the plate will begin drying out, and after a week will be too dry for most microbes to grow. Eventually, it'll become too dry for any microbe to grow, but it will become after 1 week too dry for most microbes to grow. If, on the other hand, you have humidified air, the microbes can grow for at least 2 weeks in the incubator. Sometimes you can even get 3 weeks in the incubator. And that's just because the air is humid and less moisture will evaporate out of the incubator in humid air. And that's how you just control the humidity. Just put water in a dish in the incubator, shut the door, and the heat of the incubator causes the humidity. And then the temperature, you will have a control on the incubator. You don't do it, but your instructor will set the temperature of the incubator. And it could be like room temperature, close to 25 degrees Celsius. It could be 30 degrees Celsius, and that's what the microbes on your skin grow at. It could be 35 degrees Celsius or 37 degrees Celsius. 37 degrees Celsius would be what your normal body temperature is, at least internal. You then take a look at the microbes that come out of there. And if you have isolated colonies, you can make some determination from the appearance of the colonies, like the size, the shape, the margin, the elevation. But you're not going to be able to identify the species just from inspection. You then have to do an identification, and normally that's done by further biochemical tests, which we'll begin talking about in today's lab. All right, know the terms of this lab? You don't need to go through the references. We have some videos for you to watch. Inoculation of an auger plate using streak plate culture technique, meaning um, colony streaking for colony isolation. You only need to watch this movie until 4 minutes and 15 seconds into the movie. You should also note that only 3 sectors are streaked in this video, not 4 quadrants. We then have a little video on beta hemolysis, a little video on alpha hemolysis, and a little video on gamma hemolysis. Please take a look at all of those. We have a little video on McConkey's auger. You only need to watch this video for 3 minutes and 50 seconds into the video, and then a little video on mannitol salt auger. The first thing you'll do will be practice preparing a streak plate. The virtual lab doesn't work, so it's been crossed out. And then work on the questions of the worksheet for Lab 5. Make sure I'm on the worksheet. Nope, I'm not. Just a minute here. There we go. Question 1. Describe a natural circumstance where Staphylococcus species of bacteria might benefit from salt tolerance. You cannot say something like growing on a 7% sodium chloride plate because that's not a natural circumstance. Give me a place where a Staphylococcus species grows naturally and can tolerate from exposure to salt. 2. How might hemolysis benefit a hemolytic organism? So we have a hemolytic bacteria and it can hemolyze red blood cells. How does it benefit from hemolyzing the red blood cells? 3. Imagine that you have a culture containing a single bacterial species. The culture may be one of several possible species. Describe a scenario where you use mannitol salt auger, blood auger, and EMB plates to help identify the unknown bacterial species. You must name 2 properties from each of the plate, meaning mannitol salt auger, blood auger, and EMB, to help you identify the different species. Any question about number 3? So don't tell me 2 properties for all 3 of these. You need 2 for each media. 4. What is the main goal for streaking for colony isolation? 5. What happens to the growth on the plate if you do everything correctly, but you forget to sterilize the loop in between sectors when streaking for colony isolation? You need to be specific in your answer, discussing the growth on each sector. So tell me what the growth of bacteria will be on sector 1 compared to the growth of the bacteria on sector 2, etc. Any question about number 5? Number 6, a question a little similar but different. What happens to the growth on the plate if you do everything correctly, but you transfer from the stock culture to inoculate each of the sectors? Be specific, discussing the growth you see on each sector, meaning tell me what amount of growth you see on sector 1, what amount of growth you see in sector 2, etc. Now, let me state that when you're doing everything correctly on 5 and 6, that means you are streaking the loop before you're picking up your cells from the stock culture. So don't tell me the bacteria on the plate gets contaminated because you've sterilized the loop and um, then you've picked up your cells to streak out sector 1. The thing with sector 5 is you then do not sterilize the loop in between sector 1 and sector 2. The loop is still sterile, assuming you haven't put it down or touched something to contaminate the loop, but the loop is still sterile from before Sector 1 when you did sterilize it. Any questions about 5 or 6? By the way, they do have different answers, 5 and 6. Oh, 5 and 6 are thinking questions. Oftentimes, students do not get these two questions correct on the first try, but most students do get it correct on the second try. So think about these questions and then write up your answer. 7. Suppose you successfully streak a plate and obtain isolated colonies of two different bacterial species from a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria? Be specific, and I can say thorough in your answer. Question number 8. In table below are a variety of agar plate media available to microbiologists to cultivate bacteria. To note whether each medium is non-selective media, like a general purpose media, if it's a selective media, or if it's differential media. Put it in the appropriate colony. Like nutrient agar, if you think it's selective, put an X there. If you think it's differential, put an X there. For McConkey's media, if you think it's non-selective, put an X there. And I'll give you a hint. No media is both non-selective and selective. So it should never be both non-selective and selective. It's either non-selective or selective. Is it possible it could be neither? It's possible it could be neither, non-selective or selective, meaning only differential, but not very likely. And then question 9, which agar plates were inoculated without using streak plate technique? Meaning we streaked out the cells on some of the plates here, but not on other plates. Which ones did not use the streak plate technique? All right, any question about any of these questions? I'll be here until 8 o'clock to help you with any questions or to talk about anything else you want to. If you use me before the last student logs out, because at 8 o'clock or after the last student is logged out, I'm going to log out as well. Any questions? All right, get started on the lab.